In the previous classes we have discussed different concepts in molecular biology, and in this video we are going to discuss how you will get the questions in the examinations. So the first question is, DNA fragments are option A positively charged, option B negatively charged, option C neutral, option D either positively charged or negatively charged depending on the size. So you know that here the DNA fragments are nothing but a polymer of deoxyribonucleotides. Nucleotide is mainly composed of three components: sugar, nitrogen base and phosphate. So here the phosphate is present, which imparts negative charge to the DNA molecule. So the DNA is negatively charged. So why the DNA is negatively charged? Because of the presence of phosphate. So for this question the correct option is B. DNA fragments are negatively charged. Coming to the question 2. DNA replication in bacteria occurs. Option A during S phase. Here S represents synthetic phase. Option B within nucleolus. Option C prior to fission. Option D just before transcription. So DNA replication in bacteria occurs. So the correct option is prior to fission. So you know that in eukaryotes DNA replication takes place in the nucleus and occurs during the S phase, that is in the synthetic phase. So here in prokaryotes like bacteria, DNA replication occurs continuously, not in a specific phase. And why not the second option? Here within nucleolus is the second option they have given. Nucleolus usually found in case of eukaryotes but not in case of prokaryotes. Nucleolus is nothing but a spherical ball-like structure which is present within the nucleus, so which is absent in prokaryotes. So here the first and second options are ruled out, and why not the third or fourth option, just before transcription? So there is no rule that the DNA should undergo replication just before transcription. So the correct option is prior to fission. So you know that the cell undergoes cell division. Before the cell undergoes cell division, the DNA that is present in the cell should undergo DNA replication. If the DNA replication does not take place, the cell does not undergo cell division. So you know that bacteria can divide by fission. So the correct option is DNA replication in bacteria occurs prior to fission. Coming to the third question. During DNA replication, Okazaki fragments are used to elongate. Option A the leading strand towards replication fork. Option B the lagging strand towards replication fork. Option C the leading strand away from replication fork. Option D the lagging strand away from replication fork. So whenever the DNA replication takes place, here two strands are synthesized. One strand is synthesized continuously called leading strand. Another strand synthesized discontinuously, that is called lagging strand. So Okazaki fragments are usually found in case of lagging strand, but not in case of leading strand. So option A and option C are ruled out here because you cannot find the Okazaki fragments in the leading strand. Now coming to option B and D, here the lagging B is the lagging strand towards replication fork. Option D is lagging strand away from the replication fork. As the replication proceeds in 5' to 3' direction, here the first lagging strand is synthesized towards the replication fork. So here the correct option is the lagging strand away from the replication fork. Here the question is, during DNA replication, Okazaki fragments are used to elongate from the first fragment, from the first Okazaki fragment. It takes place. It starts from the first Okazaki fragment. The first Okazaki fragment is formed towards the replication fork, so it should elongate away from the replication fork. So the correct option is D. During DNA replication, Okazaki fragments are used to elongate the lagging strand away from replication fork. Now coming to the question 4. Question number 4. The unequivocal proof that DNA as the genetic material came from the experiments of option A, Griffith, option B, Hershey and Chase, option C, Avery, MacLeod and McCarty, option D, Michelson and Stahl. So the first option is Griffith. Griffith performed the transformation experiment. Option B, Hershey and Chase. Option C, Avery, MacLeod and McCarty. Here the Avery, MacLeod and McCarty, they performed the biochemical characterization of transforming principle. Michelson and Stahl is the fourth option. He performed the semi-conservative model of DNA replication. He proved that the DNA replication is semi-conservative. So Hershey and Chase has given the unequivocal proof that DNA as the genetic material. So the correct option is B. Coming to question number 5. The association of histone H1 protein with a nucleosome indicates. Option A, DNA replication is occurring. Option B, transcription is occurring. Option C, DNA is condensed into chromatin fiber. Option D, DNA double helix is exposed. And the question is related to DNA package. So you know that different proteins are involved in DNA packaging, that is H1 protein, H2A, H2B, H3, H4. So histone octamer along with the DNA forms the nucleosome. So here the correct option is DNA is condensed into chromatin fiber. The association of histone H1 protein with nucleosome indicates DNA is condensed into chromatin fiber. DNA along with histone proteins can form the nucleosome. Here the DNA is condensed into chromatin fiber. Later it is little bit condensed into chromosome. So here the correct option for the fifth one is option C. Coming to the question number 6. Identify the correct order of organization of genetic material from largest to smallest. Option A, chromosome, genome, nucleotide, gene. Here they are asking from largest to smallest. So option B, chromosome, gene, genome, nucleotide. Option C, genome, chromosome, nucleotide, gene. Option D, genome, chromosome, gene, nucleotide. Here the genome is nothing but it is a total genetic material, the haploid set of the particular organism, the haploid set of the complete genetic material is called genome. So here the correct option is D. Here they asked from largest to smallest. Genome is the largest unit, nucleotide is the smallest unit, because DNA is composed of nucleotides. So DNA is a polymer of nucleotides. Many nucleotides can combine together to form a gene, and genes are located on the chromosomes. So all the chromosomes which combine together to form genome, haploid set of chromosomes can combine together to form genome. So from largest to smallest means genome is the largest unit, nucleotide is the smallest unit. So for the question 6, the correct option is D. Now coming to question number 7. Question number 7. The experimental proof for semi-conservative model of replication of DNA was first shown in A, plant, B, bacterium, C, virus, D, fungi. So semi-conservative mode of replication was first done by Messelson and Stahl in E. coli. So E. coli is a bacterium. So the correct option is B. So in E. coli they performed that DNA replication is they proved that DNA replication is semi-conservative. Later many scientists they performed the semi-conservative model of DNA replication in, they proved that DNA replication is semi-conservative in many organisms. So here the first proof for semi-conservative was given by Messelson and Stahl in bacterium E. coli, Escherichia coli. So for the question number 7, the correct option is B. Now coming to the question number 8. The leading and lagging strand synthesis is carried out in bacteria by option A, DNA polymerase 1, option B, DNA polymerase 2, option C, DNA polymerase 3, option D, all of these. So there are mainly 3 different types of DNA polymerases which you can find in case of prokaryotic DNA replication. So here bacteria is the prokaryote. So you know that whenever the DNA replication takes place, one strand is synthesized continuously, usually called leading strand, and the other strand is synthesized discontinuously, usually called lagging strand. In case of bacteria, the leading strand and lagging strand synthesis is carried out by single DNA polymerase. Only one DNA polymerase can carry out the synthesis of both the strands, that is DNA polymerase 3. So correct option is DNA polymerase 3. DNA polymerase 1 and DNA polymerase 2 also present in case of prokaryotes, but usually DNA polymerase 3 can synthesize the leading strand and lagging strand. DNA polymerase 3 has mainly two activities, 5' to 3' polymerizing activity, which means that they can synthesize the DNA strand from 5' to 3' direction, and it is having 3' to 5' exonuclease activity, which means that proofreading activity. If any wrong base pairs can be added, it can remove the wrong base pairs and can add the correct base pairs. So DNA polymerase 3 is usually called processive enzyme. So it is the polymerizing enzyme which can synthesize the leading and lagging strands in case of bacteria. If you are talking about the eukaryotic DNA polymerases, there are mainly three different types of eukaryotic DNA polymerases are there. They are DNA polymerase alpha, DNA polymerase delta, DNA polymerase epsilon. They can perform the synthesis of leading strand and lagging strand. Now coming to the question number 9. Griffith conducted transformation experiments on option A, Escherichia coli, option B, T2 bacteriophage, option C, Streptococcus pyogenes, option D, Diplococcus pneumonia. So you know that Frederick Griffith conducted transformation experiments on Streptococcus pneumonia. Here Streptococcus pneumonia is also called Diplococcus pneumonia or Pneumococcus pneumonia. So the correct option for 9th question is option D. Here don't confuse with Streptococcus pyogenes, that is different species pyrogenes. Streptococcus pneumonia is also called Diplococcus pneumonia. Streptococcus pneumonia is different, Diplococcus pneumonia is different. So the correct option is Streptococcus pneumonia. It's also called Diplococcus pneumonia or Pneumococcus pneumonia. So for the 9th question, the correct option is D. Now coming to the 10th question. Taylor conducted the experiments to prove semi-conservative mode of chromosomal replication on. You know that Michelson and Stahl conducted semi-conservative mode of DNA replication on E. coli. Later, after Michelson and Stahl, Taylor conducted experiments on plant. So which plant? Vicia fabaceae, type of beans plant, in which they proved that DNA replication in plants also semi-conservative. So the correct option is Vicia faba. So B. 10th question, correct option is B. Coming to the 11th question. There are special proteins that help to open up DNA double helix in front of the replication fork. These proteins are option A, DNA ligase, option B, DNA topoisomerase 1, option C, DNA gyrase, option D, DNA polymerase 1. So you know that DNA ligase mainly helps to join the DNA fragments. It doesn't have any role in open up the DNA double helix. DNA topoisomerase 1, DNA gyrase, DNA polymerase 1. DNA polymerase 1 is the name itself indicates is a polymerizing enzyme. So it cannot open up the DNA double helix. Here the DNA gyrase is the right answer. So Y means DNA gyrase usually induces negative supercoiling. Here the negative supercoiling is nothing but unwinding of DNA, that is opening up the DNA double helix. So correct option is DNA gyrase. Topoisomerases usually, DNA topoisomerase 1 usually helps to cross one DNA strand with another. It cannot relax the DNA molecule. DNA gyrase induces the negative supercoiling, which helps to unwinding the DNA, which is nothing but opening of DNA double helix. So the correct option is DNA gyrase, 11th C. Coming to the question number 12, anti-parallel strands of a DNA molecule means that option A, one strand turns anti-clockwise. Option B, the phosphate group of two strands at their ends share the same position. Option C, the phosphate groups at the start of two DNA strands are in opposite position. Option D, one strand turns clockwise. If the anti-parallel strands means one strand runs from 5' to 3' direction, another strand runs from 3' to 5' direction. That is called anti-parallel. So here in one strand, phosphate is present at one end and free hydroxyl group is present at another end. Free phosphate at one end, free hydroxyl at another end in one strand. In the opposite strand, free hydroxyl is at one end and free phosphate at the another end. So the correct option here is the phosphate groups at the start of two DNA strands are in opposite position. So 12th C. Coming to the next question, that is 13th question. Which one of the following does not follow the central dogma of molecular biology? So first of all, what is central dogma of molecular biology? So from the DNA molecule, DNA can be synthesized, that is called DNA replication. From the RNA molecule, RNA can be synthesized, that is called RNA replication. And from the DNA molecule, RNA can be synthesized, that is transcription. From the RNA molecule, proteins can be synthesized, that is called translation. So in this case, this process usually takes place in all the living organisms which is having the cell structure. So here the options they have given: A, HIV, B, P, C, Mucor, D, Chlamydomonas. HIV is a virus, P is a plant, Mucor is a fungus, Chlamydomonas is the algae. So P, Mucor and Chlamydomonas have the cell structure. HIV is not having the cell structure. It is a virus. Virus is mainly composed of only two components. They are protein coat and one of the nucleic acid, which may be either DNA or RNA. So nucleic acid and protein coat is present in the virus. And reverse transcriptase is the enzyme which helps for the synthesis of DNA, cDNA from RNA, that you can find in case of viruses. HIV. So the correct option is HIV. Virus does not follow the central dogma of molecular biology in this case. 13th question, option A is correct. Now coming to the question number 14. The strand synthesized continuously in 5' to 3' direction is option A, lagging strand, option B, leading strand, option C, Okazaki strand, option D, template strand. So whether it may be lagging strand or leading strand which is synthesized in 5' to 3' direction only, but the lagging strand is synthesized discontinuously, leading strand is synthesized continuously. Okazaki fragments are present in lagging strand which is discontinuous. So the strand synthesized continuously. The continuous synthesis you can find in case of leading strand. Leading strand can be synthesized continuously. So leading strand in 5' to 3' direction. So correct option is B, 14B. Lagging strand also synthesized in 5' to 3' direction, but it is synthesized discontinuously. So correct option is 14B. Coming to the question number 15. The circular DNA is found in A. virus, B. chloroplast, C. bacteria, D. bacteria, chloroplast and mitochondria. So circular DNA is present in bacteria, chloroplast and mitochondria. So chloroplast and mitochondria usually called semi-autonomous organelles. So the DNA present in these organisms are circular. So chloroplast and mitochondria that you can find in case of eukaryotes. So chloroplast is present in eukaryotes and also mitochondria. Bacteria also contains circular DNA. So circular DNA is found in bacteria, chloroplast and mitochondria. So option D is right answer. Coming to the question number 16. Which one of these cannot be directly synthesized from DNA? Option A, another DNA. Option B, mRNA. Option C, protein. Option D, tRNA. So DNA undergoes replication, so that the daughter DNA can be synthesized from the parental DNA. So first option is ruled out here. Coming to B, mRNA. You know that RNA can be synthesized from DNA, that is called transcription. So here whether it may be mRNA or rRNA, tRNA, whatever it may be, that RNA can be synthesized from the DNA, which is called transcription. And proteins always synthesized from the RNA molecule. Proteins cannot be directly synthesized from DNA. So for 16th question, the correct option is C, which means that proteins cannot be directly synthesized from DNA. Proteins can be synthesized from RNA. Of course, the RNA can be synthesized from DNA. So proteins can be indirectly synthesized from DNA, not directly from DNA. Coming to the question number 17. The DNA replication is option A, continuous. Option B, unidirectional. Option A, continuous, unidirectional and semi-conservative. Option B, discontinuous, unidirectional and semi-conservative. Option C, discontinuous, bidirectional and semi-conservative. Option D, semi-continuous, bidirectional and semi-conservative. So when we are discussing about the DNA replication, we know that DNA replication is semi-discontinuous or semi-continuous, which means that one strand is synthesized continuously, another strand is synthesized discontinuously. The continuous strand is called leading strand and discontinuous strand is called lagging strand. So it is semi-continuous, or you can call semi-discontinuous also. Next it is bidirectional, because here of course the both strands can be synthesized in the same direction, that is 5' to 3' direction, but the strands are in opposite direction. So it is bidirectional. and DNA replication is semi-conservative. So here in semi-conservative mode of DNA replication, each DNA molecule contains one old strand and one new strand, or one parental strand and one daughter strand. So then it is called semi-conservative. So correct option is D. Coming to question number 18. The replication rate in eukaryotic cells is about A. 50 base pairs per second, B. 500 base pairs per second, option C. 5000 base pairs per second, option D. 50,000 base pairs per second. So the replication rate is, so in case of prokaryotes, 2000 base pairs per second. The DNA polymerase can add how many base pairs? 2000 base pairs per second. 2000 base pairs means 4000 nucleotides, because each nucleotide contains one base. So, but in case of eukaryotes, how many nucleotides can be added? Only 100 nucleotides added per second, which means 50 base pairs per second. So the correct option is A, because one base pair contains 2 nucleotides. 50 base pairs contains 100 nucleotides. So 100 nucleotides are added per second in case of eukaryotes. So the replication rate in eukaryotic cells is about 50 base pairs per second. For question number 18, the correct option is A. Now question number 19. The Chargaff's rule is applicable to, you know that Watson and Crick proposed the double helical structure of DNA molecule. Based on 3 evidences, Watson and Crick proposed double helical structure of DNA molecule. Those 3 evidences are X-ray crystallographic studies, Chargaff's rule and tautomeric stability. Here the Chargaff's rule is applicable to double-stranded DNA molecule. So question number 19, the correct option is D. Now coming to the question number 20, purines are option A, monocyclic and homocyclic, option B, monocyclic and heterocyclic, option C, dicyclic and homocyclic, option D, dicyclic and heterocyclic. So purines usually consist of 2 rings. You know that pyrimidines usually consist of only 1 ring. So due to the presence of the 2 rings, the purines are dicyclic. So here option A and B are ruled out. Now coming to the option C and D, whether they are homocyclic or heterocyclic. So here the purines are made up of different atoms, usually carbon, hydrogen and nitrogen. So if a ring which is made up of a single type of atom, then it is called homocyclic. If the ring is made up of different atoms, then it is called heterocyclic. So here the purines are dicyclic and heterocyclic. So question number 20, the correct option is D. Coming to question number 21, the phosphate group is linked to pentose sugar in a nucleotide by option A, glycosidic bond, option B, hydrogen bond, option C, ester bond, option D, peptide bond. So you know that nucleotide is mainly composed of 3 components. They are sugar, nitrogen base and phosphate. Here hydrogen bonds are present between purines and pyrimidines. Ester bond is present between sugar and phosphate, we already discussed in the concepts. Peptide bond is present between the amino acids. So here the correct option is A, glycosidic bond. Sugar and phosphate. Sugar and phosphate are linked by ester bond. Sugar and base is linked by glycosidic bond. Sugar and phosphate are linked by ester bond. So once again, question number 21. The phosphate group is linked to pentose sugar in a nucleotide by. Option A, glycosidic bond. Option B, hydrogen bond. Option C, ester bond. Option D, peptide bond. So peptide bond is present in the proteins, which means amino acids are linked by peptide bond. Hydrogen bonds are present between the purines and pyrimidines. N-glycosidic bond is present between sugar and nitrogen base. Here sugar and phosphate is linked by ester bond. So correct option is C. Ester bond is the right answer. Now coming to question number 22. A bacterium grown over the medium having radioactive sulfur incorporates radioactivity in. Option A, carbohydrates. Option B, proteins. Option C, DNA. Option D, RNA. So radioactivity recovered in or incorporates in. So they used here radioactive sulfur. So phosphorus is present in DNA, absent in proteins. Sulfur is present in proteins, absent in nucleic acids, that is DNA and RNA. So here option A, C, D are ruled out, as the carbohydrates does not contain sulfur. Even DNA, RNA also does not contain sulfur. Sulfur is present only in proteins. So radioactive sulfur incorporates its radioactivity in proteins because the proteins contains sulfur. And one more thing here, there are amino acids which are rich in sulfur, usually present in proteins. They are cysteine and methionine. Cysteine and methionine are the amino acids which are rich in sulfur, usually present in proteins. So correct option is B, 22B. Now coming to the question number 23. cDNA is formed by. Here the C represents complementary. So complementary DNA is formed by. Option A, replication. Option B, transcription. Option C, reverse transcription. Option D, translation. So synthesis of daughter DNA from parental DNA is called replication. So option A is ruled out. Synthesis of RNA from DNA is called transcription. That is also ruled out. Option D, translation, which is nothing but synthesis of proteins from RNA is called translation. So that is also ruled out. So reverse transcription is the right answer. So what is reverse transcription? Synthesis of complementary DNA from RNA is called reverse transcription. So whenever you can find the cDNA, which means it is synthesized from RNA molecule. So the correct option is C, 23C. Now coming to the question number 24. How many nucleotides will be present in a DNA of 20,000 base pairs? Option A, 40,000. Option B, 4,000. Option C, 20,000. Option D, 2,000. Here they asked how many nucleotides will be present in a DNA of 20,000 base pairs. One base pair contains two bases. which contains 2 nucleotides. So one base pair contains 2 nucleotides. So 20,000 base pairs contains 40,000 nucleotides. Each nucleotide contains 1 base, 1 sugar, 1 phosphate. Each nucleotide contains 1 base, 1 sugar, 1 phosphate. So each nucleotide contains only 1 base. So one base pair contains 2 nucleotides. 20,000 base pairs contains 40,000 nucleotides. So option A, 24th question, option A is the right answer. Now coming to 25th question. Teminism is option A, reverse transcription, option B, DNA replication, option C, RNA replication, option D, transcription. So reverse transcription is the process that is discovered by a scientist named Temin. That's why this reverse transcription process is also called Teminism. So what is reverse transcription? The synthesis of cDNA from RNA is called reverse transcription. So 25th question, option A is the right answer. Reverse transcription is also called Teminism. Coming to the question number 26. Pyrimidines are A, monocyclic and homocyclic, option B, monocyclic and heterocyclic, option C, dicyclic and homocyclic, option D, dicyclic and heterocyclic. So pyrimidines usually consist of how many rings? Only one ring is present in the pyrimidine. But purines consist of 2 rings. Pyrimidines consist of only 1 ring. Hence the pyrimidines are monocyclic. So option C and D is ruled out here. So pyrimidines are monocyclic. Whether they are homocyclic or heterocyclic. So pyrimidine is also made up of different atoms like carbon, hydrogen and nitrogen. So it is heterocyclic. Homocyclic means, I already said, made up of only one type of atoms. Heterocyclic means made up of different atoms. So pyrimidines are monocyclic and heterocyclic. So option B is the right answer. Now coming to the question number 27. Sugar and pyrimidine is linked by option A, N-glycosidic bond, option B, ester bond, option C, hydrogen bond, option D, peptide bond. So we already discussed this pyrimidine is nothing but nitrogen base. Here sugar and nitrogen base is linked by N-glycosidic bond. So ester bond you can find in case of sugar and phosphate. Sugar and phosphate is linked by ester bond. Purines and pyrimidines are linked by hydrogen bonds. Amino acids are linked by peptide bond. Nucleotides are linked by phosphodiester bond. So here the sugar and nitrogen base, that is nothing but pyrimidine, is linked by N-glycosidic bond. So C1, pentose sugar consists of 5 carbons. Here among the 5 carbons, the N-glycosidic bond you can find in C1 carbon, that is C1 is involved in the N-glycosidic bond. C1 of the sugar involved in the N-glycosidic bond, and N1 of the pyrimidine also involved in the N-glycosidic bond. So C1 of sugar, N1 of pyrimidine are involved in N-glycosidic bond. So the correct option is 27th question, A, N-glycosidic bond. So these are the few things that we discussed in this class. The remaining bits that we will discuss in the next class. Okay.